leads me to our two invited guests today, Dr. Andrew Leach and Dr. Paula Magariños. So I want to introduce you guys really quick, and also thank you so much for presenting. It's really an honor to have you. Just to say that the EBI and Andrew and the team has been like a partner in IDG for a long time. A lot of the SureChEMBL resources are incorporated into the main knowledge portal Pharos of the IDG. And there was also a lot of work to extract information from patents that is now also available. And that was, there was a lot of work was done about understudy targets related to the IDG by you guys. So it's really a partner in the program like we could only wish for. So a brief introduction. So Andrew Leach is the head of chemical biology and head of industry partners at SureChEMBL EBI at the Wellcome Genome Campus in Kingston, UK. And he has professional expertise for over 25 years in academia and large pharma. He joined EBI in 2016 after a wide-ranging career in industry drug discovery where he held multiple roles. Those included leadership and management of large multinational groups in computational biology, structural biology, and biophysics. And I think, you know, everybody who knows Andrew knows he is like a world-recognized expert in structural biology and computational chemistry. He also led the target selection, hit and lead discovery activities for multiple target families, including proteases, ion channels, and epigenetics targets. He led the development and subsequent implementation of global discovery platforms, including fragment-based drug discovery, phenotypic screening, and his diverse range of expertise have provided him with like deep knowledge and practical drug discovery expertise and understanding of the key roles of data and informatics in decision-making. Yeah, and that led him to lead the chemical biology effort, the SureChEMBL effort at the EBI with more than 20 people as staff. And of course, you know, Square is PI on many grants, and you can look up long publication records, you know, that's extremely significant. So the other speaker is Dr. Paula Magariños. I'm sorry if I messed up the name. So she is a bioactivity data manager for the chemical biology team at SureChEMBL EBI. And she's an expert in software development, bioactivity data curation, and drug information sources to develop workflows that can be used to efficiently identify and extract data and information from relevant sources. So as you guys know, the SureChEMBL resource, you know, it's a huge effort to extract information from many documents, including journal publications and patents. And she is, you know, one of the individuals who actually makes it happen. Her PhD is from Universidad Nacional de San Martín in bioinformatics, and before that she has a degree in biology and life science from the University of Buenos Aires. So thank you guys so much for presenting, and we really look forward to your presentation, chemical biology resources at the EBI, current status, and some future plans.
Excellent. Thank you very much, Stefan. Thank you, Anna. So the plan for today is shown on this slide here. I am going to give you what I'll say up front is a fairly high-level overview of some of the chemical biology resources at the EBI. I'm anticipating that quite a few people on the call are users of our resources, which is great. And in some ways, this session is as much for you to give us feedback and comments and thoughts as much as for me to sort of tell you some of what we're doing. So I'm going to lift the lid a little bit on, you know, what goes on behind the scenes within the team to deliver these databases, and also some of our emerging plans for the future, again, of which I'll be very grateful and pleased to hear your thoughts and comments. And then I'm going to hand over to Paula, who, as Stefan mentioned, is going to go a bit deeper into the work that she's been doing as part of the IDG project and program to identify patterns from which we can extract useful bioactivity data that helps to illuminate some of the hitherto dark targets that have been identified as part of the project. Okay, so just a little bit of high-level background on the EBI for those of you who are not familiar with our institute. So we're part of the European Molecular Biology Lab, that's EMBL, and EMBL has 6 sites, of which the site in Hingston, where we are based, is one. And at the EBI, we're a completely in silico sort of institute. We don't have any laboratory, any labs within the institute there, although we are co-located with the Sanger Institute, which obviously has the heavy-duty sequencing and other facilities available to them. And in terms of data resources, there's actually a large number of data resources. Again, I'm sure many of you are users of one, if not more, of these. And it's a really sort of commercial operation in the sense that we get, huge numbers of hits. We have a very sort of large and robust compute infrastructure. We have a large number of staff that continues to grow at a tremendous pace. We're very multinational, international. So that's just hopefully giving a little bit of a flavour of the EBI. And again, anybody who's in the UK and is in the Cambridge area, do feel free to reach out and see if you want to come pay us a visit. So here are some of the data resources. This slide keeps changing all the time, so it's always a little bit out of date, but hopefully it gives you a reasonably sort of up-to-date view of the different resources that we have across the different sort of areas. I'll just highlight the AlphaFold database. Obviously still quite a very sort of cutting and newsworthy development, especially with the most recent release that massively expands the number of predictive structures available. But then again, we have a number of resources that go back many years, if not decades. The PDB-E as part of the wider worldwide PDB being a good example, along with Ensemble and Uniprot and others. So my group looks after chemical biology. These are the 4 key resources that we deliver, of which I'm going to focus today very much on SureChEMBL and SureChEMBL, but just for those of you who perhaps are interested or maybe even are users of KEBI, for instance, this is actually the longest established chemical biology resource at the EBI. It goes back to 2004, very much developed for the sort of biology community. It's a user request driven resource. It's relatively small compared certainly to SureChEMBL and SureChEMBL, but it's completely human curated. And a key feature of KEBI is the ontology, which is integrated into and widely used by the ontology community. So resources such as Go, for example, are critically dependent upon KEBI for the chemistry knowledge that they incorporate. But as I said, I'm going to focus today primarily on SureChEMBL and SureChEMBL, which are a little bit more recent, although SureChEMBL, we recently, well, a couple of years ago, we celebrated the 10th anniversary of SureChEMBL, and we had a little symposium on site, which was great to be able to invite and welcome a number of our sort of wider user community to that. But just in terms of other resources at the EBI that I reference or include data on small molecules, actually quite a number of these. Metabolites is our metabolomics archive, and that uses KEBI as its sort of structure register. The PDB obviously contains much information on protein ligand structures, and actually the PDB has quite a sophisticated structure aware searching capability that allows you to query that resource more detail around the small molecules that it contains. And then there are other resources, some of which I will come back to later in the talk, particularly around the Biogenome Archive, which is a much more recent resource. One thing that certainly I continue to be keen to do is to integrate the chemistry resources at the EBI perhaps more comprehensively than they are. It's perhaps a natural consequence just of the way that sometimes these resources evolve and develop, that they sort of tend to only start out kind of on their own, but certainly now I think we can see some opportunities to consolidate, and indeed we are consolidating further, certainly behind the scenes on the infrastructure, on some of the underlying capabilities. And this is something that I think we have still progress to make, but nevertheless, we're embarking on that journey. SureChEMBL then. So SureChEMBL is a database that is focused around bioactive compounds and drug data. We have just released version 31, which is the most recent one of those in July. On the right, you can see the sort of high-level summary of the number of data points we have. And the bulk of, or a lot of the data in SureChEMBL comes from curation from the literature. So if we take this paper as an illustration, this is a JMedChem paper. Every single piece of bioactivity data and every structure will be extracted, curated into SureChEMBL, whether it's data against the target, data against some sort of cell-based assay, physicochemical data, in vivo data, everything goes into SureChEMBL. That's something that is a key feature of SureChEMBL, that really we are broadly looking at drug discovery data. And the other key aspect of everything we do is that really ensuring as far as we can the quality of what goes into SureChEMBL is the number one priority for us. And this slide just illustrates, I'm not going to go into the details, but just illustrates all the different sort of steps that one needs to go through, or some of the steps, it's not everything here, by way of standardizing, checking, confirming the validity, the accuracy of the data that we put into SureChEMBL. Now, we don't get it right all the time, and indeed we sometimes pick up potential errors actually in the primary literature, which we will flag. And again, that's one aspect of SureChEMBL that is always worth bearing in mind, that we do endeavor to, where we can, provide some sort of clue or hint as to whether we think that particular value is believable. But nevertheless, that is part of the overall process, and that allows us to then feed this data and integrate it into other resources using relevant ontologies, vocabularies, and identifiers. SureChEMBL continues to grow. You can see here the changes in the numbers of targets, compounds, activities, and underlying documents. And I think for me, as somebody with a drug discovery background, then the most important aspect of SureChEMBL from a user point of view is that people actually are using it to do real scientific research, to answer their science questions. And we know that not everything that people do with SureChEMBL is published, but it's great to see that people do publish the use of the resource, and the number of citations to, I guess, the key sort of landmark papers to SureChEMBL continues to grow, which again is great to see. Another element of what we do, and increasingly so, is to both use and contribute open source software. So the original SureChEMBL was built partly on commercial software, and over the years we've been trying to move more to open source tools, certainly as those become more available and their quality is of the standard that we need. And as part of our activities, where we can, we look to contribute back. So just some examples here, we have worked with R.D. Kitt and Greg Landrum in particular to develop a chemical structure standardization process. And one of my colleagues, Eloy Felix, has developed a package for fast molecular similarity searching, which again is part of what SureChEMBL is using, and indeed other of our resources. And both of these, together with other tools and developments, are available for download and reuse as part of GitHub and GitLab. One of the challenges that one often always faces as part of a sort of living and breathing data resource is that there's this balance between adding and enhancing the resource by adding new features and making that sustainable within the constraints of the resources and the funding that we're able to obtain. And one of the, again, things that we've been doing sort of gradually over the last few years is sort of thinking hard about, where are we now? Where are we going? Are our users actually using all of the sort of features that sometimes we've added, often, for very good reasons at the time, but perhaps over time are not so heavily used? We can, as many others resources will do, you can do a certain amount using, for example, the tools that come with a web interface, but one other thing that we have done ourselves, and which actually is fairly common at the EBI, is every so often to seek direct feedback from our users. And so one thing that we did last year was run a user survey. We had, I think, what generally is a pretty decent response in terms of the numbers of users that commented and fed back. And whilst this isn't the only mechanism that we use, nevertheless, it is a useful pointer perhaps to what are some of the features that, especially those which are time-consuming, resource-consuming to maintain, but which perhaps are less widely used. So this is illustrated here. It was quite interesting actually for us to see that the drug information in SureChEMBL, which perhaps it isn't as well known as certain other resources, but which again we put quite a bit of effort into, is actually growing in popularity. As well as, again, there are certain sort of features that perhaps are less heavily used, and which we will certainly continue to look at with due warning. And we do try to give a good heads up in terms of any changes that we're planning to make before we deprecate a feature. But these are just some of the areas where we'll continue to sort of look at as part of a living, breathing resource that, again, we're looking to be as useful as we possibly can to the widest number of users. So just then on that topic of drug, but also clinical candidates. So we particularly, these obviously are compounds, not only compounds actually, they also include protein therapeutics, but these are entities for which there's often a rather more data available than is the case for a typical structure in a JMA Chem paper. And so in the work that we do to annotate drugs and clinical candidates, then we use a variety of resources you can see listed on the left here. And there's a number of key properties and features that we're looking to identify. So one obviously is, the sort of underlying basics, the name, the structure. Other elements around, well, what, is that molecule for? How's it administered? But a key thing is, what's the underlying biological mechanism by which that therapeutic is having its effect? And what are the diseases for which it is being used? More recently, we've expanded this to include information on drug warnings. And this, again, is something that we published a paper on a little while ago. And it's an area we continue to maintain and keep up to date. One of the particular aspects of the mechanism of action data is that there's a growing number of cases where the target is some sort of variant. Now, this clearly often arises quite frequently in particularly oncology and in infectious diseases. But we also are sort of seeing this popping up in other cases. And these are situations where, again, we really dig as deeply as we can into the available information. We're cross-referencing this against UniProts, looking to indicate where in the actual sequence is the variant. And there's actually quite a large, several thousands of examples of this particular type of data now in SureChEMBL, which, if you're interested in variants and variation, then I'd certainly encourage you to go and take a look. One illustration of the ways in which you can then use both the drug and more broad bioactivity data in SureChEMBL is this piece of work. This is, I guess, more of a research project that we worked on with Paul Leeson, who has himself a long history in drug discovery in several companies, is now a consultant, but whom I've worked with in the past. And in some ways, this is a somewhat age-old question, what makes a drug special? And I guess what we were looking to do was, be as definitive as possible, a set of both drugs with their known targets, together with what we call discovery compounds or comparator compounds. So these are compounds in SureChEMBL with a measured experimental bioactivity against that target that we can then use to sort of see, what is different? Is there anything that's different about these drugs compared to these discovery compounds? So this is work that was published last year. So I'll kind of go through it fairly quickly, but this is really just to give you a taster of really quite a lot of work, and I should give due credit to Paul, because he really did a lot of the actual analysis and created these very nice sort of graphic plots and so on. As we, I think, have seen, those of us who have been in drug discovery for a while, you know, then the field has sort of picked up on some of the learnings that came out, for example, around Chris Lipinski's seminal work. And I think it's fair to say that, you know, most of the compounds that we certainly are seeing today, by and large, you know, conform to the sort of Lipinski type rules and other similar properties. But what we did observe is that still, you know, ligand efficiency remains a key property, a key aspect of that does look as though it differentiates the sort of drugs that are making it through from those compounds, be it the ligand deficiency normalized by molecular size or the lipophilic ligand deficiency. And again, there's some nice examples in the paper that Paul shows where, you know, you can actually see this played out in the particular case histories that one can look at. So again, I sort of encourage you to look at that paper and that data set if you're interested. The data set's available. And indeed, we are ourselves using this with other collaborations that I won't say anything about today, but which hopefully we'll be publishing in the near future, you know, to look at other aspects of, you know, drugs and clinical candidates, because again, this is work that Paula herself particularly works on to identify, you know, those later stage compounds that, you know, have made it through into the clinic and which therefore clearly have some sort of special characteristics about them that that deem them able to be taken that far. One of the recurring themes that again comes up in the context of bioinformatics and cheminformatics resources is, well, you're putting all this human effort into curating this data, and clearly that is time consuming, it's expensive. Can you automate it? And that's a very good question. And we, and indeed my other colleagues, are increasingly looking at, you know, how we might be able to incorporate some of the more recent, you know, natural language processing and other type of methods into the work that we do. One example of a project that we ran, this was actually with Open Targets, but, you know, it's sort of generally applicable to SureChEMBL, is looking again at this question of drug indications and mechanisms of action. In this particular case, looking at the drug labels that can get out of Daily Med. And as you can see at the bottom, you know, can we process and analyze, you know, the text that is contained within these labels to extract sort of relevant information and at least make some sort of progress towards automating this particular aspect. There's several thousands, if not tens of thousands, of these sort of labels that, you know, are available, and they actually do contain some quite valuable information. The bottom line is that as part of as a research project, then indeed we were able to build you know, quite a sophisticated machine learning model that had quite a reasonable performance. And indeed, we did use it in order to process, you know, quite a number of these labels. On the other hand, it's not by any means 100% foolproof. There's always cases that you find which, you know, are wrong and inaccurate, and that then creates a bit of a dilemma for us, which is, you know, well, what exactly is the role of these sorts of methods? And I think we're still feeling away a little bit in terms of, you know, how to get that sort of combination of human expert and sort of machine approach to get the most efficient whilst retaining the kind of quality that we're looking for. But nevertheless, and there are, again, some other sort of practical issues with this that just relate to the complexity of, you know, drug formulation, drug structure representation, and so on. But nevertheless, I think, you know, definitely from our point of view, this is an area we continue to be interested in, continues to work on. Again, I know there's other groups from IDG that are also working in this sort of area, perhaps looking at other types of data. And I think certainly over the past, you know, 5 years or so, we've seen quite significant developments in this field, which hopefully we'll be able to capitalize upon as we go forward. Okay, so that's a little bit of a summary of what SureChEMBL is, some of the things we've been doing more recently. Again, Paula will say more detail some of the work that she's been doing with patents, but I just want to sort of introduce some of our sort of thoughts for the future. So again, this is very much where comments and feedback from this group will be of interest, particularly around the data, the content. So SureChEMBL, maybe not everybody's aware, but we basically have a core set of mostly medicinal chemistry journals from which we extract and curate pretty much every paper, because they all typically contain data of interest. So the classic example would be Journal of Medicinal Chemistry. But I think certainly over the years since sort of SureChEMBL became a public resource, then we have seen chemical biology data published in other journals and in other forums. And I think one question is, where else might we go? And we do have mechanisms for identifying, again, potentially interesting papers, but we're always keen to hear from others. But also, I think there's an interesting question as to what data is actually adding most value, because I think it's probably undoubtedly the case that even if we could identify every single piece of bioactivity data, we wouldn't have the resources or the budget to process all of those. And so there is this question of well, what is most value? What adds the most value? I think the e-IDG project in this context is very interesting and very useful, because by defining a set of quotes dark or underexplored targets, then that clearly is a great sort of starting point. And you might well then prioritize more highly, early bioactivity data on a unexplored, underexplored target, compared to a very well-worked, very well-studied protein for which there's a lot of data already in SureChEMBL. And we're working with, for example, the PDBE team in a project called BioChemGraph, along with the Cambridge Crystallographic Data Center, to look at the data and the sort of protein ligand structures, and just to see what are the ways, for which of those is there bioactivity data reported, where is it, and how might we then enhance, for example, that particular data set. Another growing area for us is depository data sets. Historically, we have particularly worked with those working in the area of developing world diseases, where there's been a desire to make the data public, and SureChEMBL has been a good sort of vehicle for doing that. But increasingly, we're working with what I call strategic partners, and again, interested to hear anybody on this call, where groups that maybe are generating or routinely generating quite large amounts of data, where we perhaps can work with them to establish a good workflow that enables a sort of continual, sort of seamless process of depositing data into SureChEMBL, as part of the grand vision will be towards some sort of universal deposition process. I think it's fair to say that compared, for example, to protein structure or to DNA sequence, then chemical biology doesn't really currently have that kind of culture of open data. Again, we can go into the reasons for that, but clearly one of the reasons is that the data we work with is very complex. It's very broad, it's very diverse, and so it's actually quite complicated to sort of come up with a truly universal process. But I don't think that should be the reason not to at least aspire to move in that direction. And then another area that, again, I think we're seeing increasingly is more complex data. And here, what we're not going to do is to sort of make SureChEMBL the repository for all different data types. That doesn't make any sense at all. What we will do, though, is to work with, for example, other colleagues at the EBI who may have data of different data types, and look to capture the appropriate data type in the appropriate database, and then obviously to integrate it. So that is, I'll give you an example of that in a minute, which comes from one of the projects that we are involved in. This is a European IMI project called UBOpen. The Structural Genomics Consortium is the lead partner in this group, but there's others from both academia and industry. And these are the headline goals. So it's pretty ambitious, looking to create an open chemogenomics set to deliver chemical probes in new areas, to develop new human tissue assays, and critically, all of the outputs and data will be made open access. And the role that we're playing is to work to develop and deliver the public gateway that will be used to as a sort of landing page for people who want to access the data. But we're also working with the team and with other colleagues at the EBI to again establish these workflows for getting appropriate data into SureChEMBL or other places as appropriate. Just one example of that we're just on the verge of making public is what we call multiplex data. So this is data coming from a sort of various phenotypic assays, image-based assays that are reporting on the sort of cell health or the sort of viability of cells when dosed with the compounds. This is quite complex. There's multiple time points. There's different readouts. There's different assay types. And here, for example, the images go into Bioimage Archive because that's where they should sit. But in SureChEMBL, we have sort of summary data that represents the sort of conclusions coming out of those assays, which then feeds into the UB Open Gateway. And what we're looking to do is to develop ways that then the user can navigate and interrogate this data, hopefully in useful, interesting ways to understand, what's going on through this combination of multiple resources. So I see this as an area that will grow in the future. Okay, so moving on then, sort of as a bit of an intro to Paula's section. So SureChEMBL is another resource that we look after. It contains patent data. It's very large, so it's fully automated. There's no human curation involved here. And again, it particularly from our point of view, we look to identify chemical structures in patents together with various other annotations. SureChEMBL's history is quite interesting. It was a sort of semi-commercial system before it was acquired by the EBI, and that's kind of largely the system that we've been making available. And I think maybe not so much for this call, but certainly my observation will be that patent data on the whole isn't very widely understood and used, certainly the academic community. So one of our goals with the SureChEMBL project is to raise the awareness of information in patents. At the bottom here, we've identified a number of different use cases that we see as examples of the kinds of queries that you might wish to do using patents. And I think the other point, again, this maybe not be so new to users on this call, but there's actually a lot of valuable data in patents, which either appears after quite a time lag in the literature, if it appears at all. So again, Paul will say a little bit more about that. We have recently been awarded some funds from the Wellcome Trust to basically completely overhaul the infrastructure. So for those of you who are SureChEMBL users and perhaps have been a little bit frustrated recently about its accessibility and so on, we are working hard to really develop and deliver a much more sustainable, supportable and modern system that will have, a nice newer UI, but critically will be a lot easier to support on the back end, together with a sort of a public API, which again I know many people have been looking for. So I'm going to stop at that point. I'll hand over to Paula, and Paula, if you just want to say next slide, then we can do that to progress things.
Thank you. Yes, so I'm going to talk about my work about searching for targets and bioactivity data in patents. So as Andrew mentioned, SureChEMBL is a very big database. The number of patents that are processed every day and the number of compounds is very large. So the goal of this work, one of the key points of this work was to try to find ways to prioritize the patents in order to find the patents that would be more useful for what we wanted. That was to try to find patents with bioactivity data on the IDG TDARC antibio targets. So this is an example of a patent with bioactivity data. In this case, is for GPR6, which some years ago was a T-bio target. And this patent has a number of compounds in the text, and then it has tables like the one shown in this slide, where for each compound there is a measurement on the target. So this is the sort of patent that we are looking for. Next, please. So as a first step, I tried to search, so I downloaded the XML files of the patents, and I tried to search for the tables within the files containing these bioactivity keywords that are shown here. And I flagged all the patents that had this kind of tables with these keywords as potentially having bioactivity data. By doing this, 2% of the patents could be flagged. So it is one first step that narrows down a bit the list. Next, please. On the other hand, we wanted to find information on the understudied targets. So I tried to search for these targets, looking for phrases that are shown here that could indicate that the patent had bioactivity data. So for each patent, I registered which phrases were found for which target and how many times they were mentioned and in which section. So in the title, the abstract, the description, or the claim section of the patent. Next one, please. The first time I did this, I chose to search patents from 2012 to 2018 that were flagged as life science relevant. This flag is an internal flag in our database that is related to the presence of certain IPC codes that are codes that describe the patent according to the area of application of each patent, such as electronics or chemistry or medicine, etc. There are many. It's a very long list. So this group of patents were 3.7 million patents as a start, and I ran these two processes that I described before. So first, the search for tables with bioactivity keywords, and separately looking for the targets. And according to the output of each of these, I could classify the patents into 6 groups. And then I read patents from each group to see which groups were more likely to have data. Of course, I expected that the data would be mostly in the group 1 because these are patents that have tables, so they were flagged as potentially having bioactivity data, but also they mentioned the targets in the title or abstract. And indeed, this was the group with more, the higher proportion of data. But I also found data in the group 2 and 3, so the patents with tables and with targets mentioned in the description and claim section, and the patents that didn't have tables but had titles, targets mentioned in the title or abstract. There was a bit of information in the group 4 and almost nothing in the groups 5 and 6, which was expected. So after reading patents from each group, we sent 226 for curation, for manual curation and extraction of the bioactivities. And so when we got the results back, it turned out that 197 patents were positive and 29 were negative. Negative here means without information on IDG, T-dark, T-bio targets, but they might contain information on other targets, and everything gets extracted, not only the IDG target. So when we send the patent for curation, everything that is there is extracted. And then when we get the results back, we have another round of curation in the SureChEMBL group, because this information needs to be mapped to SureChEMBL targets. and in some cases, some targets need to be created if they don't exist. And finally, after this round of curation, this goes into SureChEMBL and is eventually picked up by e-IDG when there is a new release of the TCRD database. So this is the whole process. Next one, please. So these are some examples of targets for which there was data. And mostly kinases and GPCRs. And here there are also some numbers of compounds found. And here compounds mean small molecules because so we found some patents that have information on, for example, antibodies or other types of molecules such as RNA, but these are not considered here. So I'm not considering positive a patent that has information on antibodies or RNA, only small molecules. Next one. So this is one example, again, for GPR6. This is data taken from SureChEMBL that shows that the first molecule for this target comes from a patent. And then some years later, there is another molecule from patents that has a value of bioactivity that is very, is much smaller than the cutoff, the e-IDG cutoff for the GPCR family. And some years later, there is one molecule from literature. So this is one case where the information seems to appear first in patents, or some years earlier in patents than in literature. Next one. So far, after doing this first round of curation and extraction of bioactivity data, we now have some positive and negative data set. So the next step was to see if we could use this positive and negative data set to apply some machine learning methods and try to prioritize the patents in a different way that didn't involve so much reading of patents. So I tried to train two naive-based models using scikit-learn library. And then I tried to apply these models to search patents from 2019, 20, and 21. Yes. So for this, I divided the problem in two parts. First, given a patent for which I can find targets mentioned in the phrases that I was looking for, the first challenge is to determine which of these targets are the true target of the patent, meaning the target for which there is data, because each patent is generally very long and they can mention a lot of targets, but generally there is data for a few of them. So this is the first step, to determine which is the target of the patent. And here in this slide, I'm showing a patent for which there are two targets found that are shown in the match column, but only one has data. And then the second part of the problem is to see if a patent is likely to contain bioactivity data of small molecules for a particular target. And an example of this would be a patent that has as a target an e-IDG target, but that is describing antibodies for this target. So this wouldn't be a positive patent. The next one. So as I divided this problem in two, I had to train two models, one for the target and one for the patent. For the target model, I used as features the phrases that were found in each patent, how many times and in which sections of the patent. And for the patent model, I used first the output of the target model, so whether or not a particular patent has a target predicted for that patent. And then also other features such as the presence of tables with bioactivity keywords, the presence of tables even if they don't have bioactivity keywords, the presence of compounds in the patent, the presence of new units, bioactivity units mentioned in the text. And finally, the presence of keywords related to antisense or antibodies or biomarkers that are the patterns that we don't want. Next one. So first I tried to apply these models and to predict the patterns from 2019 to 2020. And I use as target list the shorter IDG list that can be found in drug-of-the-genome.net, the one from July 2019 and November 2020 for 2019 and 2020 patents. And I found 47 patents that were sent for curation. 37 of these patents were positive, so they had data against IDG targets. 10 were negative, so they had data against other targets that were single proteins that were not in the IDG list, and also data against cell lines, protein complexes, and protein-protein interactions, which are other target types in ChEMBL. These patents had data against 116 IDG targets, of which 85 kinases and 40 PCRs have bioactivity data within the IDG cutoff. And these results are in ChEMBL 31, which was released in August this year. And for the 2021 patents, I decided to use to use the full TDARC TBio list as of March 2022. And then because for the 2019 and 2020, I had used the shorter lists, I decided to run this again against 2019, 20, and 21 for the full IDG TDARC antibio list. And I found 82 patents that were sent for curation. And so these are results that we have already back, but because they haven't been loaded and the targets haven't been mapped to ChEMBL targets, I still don't have the numbers of this new batch, but it's likely that this will be included in ChEMBL 32. So as future and ongoing plans, we plan to continue with the bioactivity data extraction, improving the machine learning models, which are at this point preliminary work, and I think they can be improved a lot. And also with each round of curation and of each time we send patents for curation, we have more data that can be used for the next round of patent search. And we also have some questions that we think that they could be interesting to see if they can be answered with patents. For example, finding the oldest patent for each drug and see if we can identify the target, and this could have a potential application to clinical candidates where we might not know the target. And also, for example, another question would be, can we identify the disease from the earliest patent? So that's all the work.
Great, excellent. Thanks, Paula. So I just finish by thanking not only Paula, but actually the entire team, together with many former colleagues who've now moved on, some of whom are on the picture on the right, and